Okay, so we are going to look at glycolysis, which is one of the first steps in the process of respiration. Glycolysis enables the conversion of glucose into pyruvate, and this process occurs in all living organisms. It occurs basically in the cytoplasm of cells, and it involves a number of enzymatic reactions, which ultimately result into production of at least of two molecules of pyruvate. It is an efficient way. Glycolysis is an efficient way of producing energy, where we produce at least a net of two molecules per glucose. The number of we produce a net of two ATP molecules, but generally in total we produce four ATP molecules. But in the process we use two, such that we have a net of two ATP molecules per glucose molecule. The process does not require oxygen, so we can say it is oxygen-independent pathway. The other feature is that glycolysis produces metabolic pathways, metabolic intermediates that can be used in other pathways, such as the synthesis of lipids and amino acids and nucleotides. Glycolysis can be, or glycolysis actually takes place as a series of reactions that are 10 in number. Okay, there are 10 steps of glycolysis. We can divide them into two phases. Okay, the first 5 steps belong to the energy investment phase, which basically involves the utilization of energy. That's why we call it energy investment phase. And then the second phase is the energy payoff. This is now when we generate the 4 ATP molecules. So the first step of glycolysis involves the conversion of glucose into glucose 6-phosphate. This is an important step because it is actually irreversible and plays an important role in the regulation of glycolysis. The enzyme involved is called hexokinase, and therefore, because it involves the addition of a phosphate group, we use the phosphate group from ATP. So therefore, ATP is being utilized at this stage. The next step involves glucose 6-phosphate being converted into fructose 6-phosphate through a process of isomerization and the enzyme phosphohexokinase, isomerase is what is actually involved. And then the fructose 6-phosphate is now converted into fructose 1,6-biphosphate using an enzyme phosphofructokinase 1. So it basically adds another phosphate group such that we have 2 phosphate groups. So such that we get fructose 1,6-bisphosphate. Okay, and then this compound now is cleavaged to produce 2 molecules, each one having at least having 3 carbon atoms. Okay, each one having 3 carbon atoms. The enzyme involved is what is called aldolase enzyme. Okay, so the two compounds are dihydroxyacetone phosphate and then glyceraldehyde 3 phosphate. Now, the last step of the first half of glycolysis is the interconversion of uh, dihydroxyacetone phosphate into glyceraldehyde 3 phosphate, and also the conversion of glyceraldehyde 3 phosphate into dihydroxyacetone phosphate. So there is this irreversible interconversion between uh, from one molecule to the other. Now, moving into the second phase of glycolysis. The first one is the conversion of glyceraldehyde 3 phosphate into 1,3-bisphosphate bisphoglycerate. Okay, this conversion is catalyzed by the enzyme glyceraldehyde phosphodehydrogenase. Basically, hydrogen atoms are being removed. Okay, and then a phosphate group is being added such that we have 1,3-bisphosphoglycerate. Okay. The next step now involves the removal of the phosphate group. In the process, ATP is actually made. Since two molecules are involved, or we have two molecules, there are two ATP molecules are produced at that stage, such that we have 3-phosphoglycerate molecules. Okay, 3-phosphoglycerate. Then these are two molecules. Another step involved there is the transformation of this molecule to form phosphoglycerate, 2-phosphoglycerate. And these are basically two molecules also. And phosphoglycerate mutase is the enzyme responsible. The next step is the conversion of 2-phosphoglycerate into phosphoenolpyruvate with the help of one enzyme, enolase. The last step is the conversion of the phosphoenolpyruvate into pyruvate, and this process also gives us ATP molecules, two of them, because two molecules are actually involved. And the enzyme involved is pyruvate kinase. So the pyruvate molecules, to the two pyruvate molecules, actually, they are the major products of glycolysis, and they can be utilized depending on the absence or presence of oxygen. If there is oxygen, pyruvate is converted into acetyl-CoA by an enzyme called pyruvate dehydrogenase complex. And then the acetyl-CoA now can enter into the Krebs cycle to generate more energy and reduce the NADH and then FADH2 molecules. In absence of oxygen, pyruvate is converted into lactic acid, which can be taken to the liver. So therefore, we produce 4 molecules of ATP from glycolysis. Because we use 2 in the process, we end up with a net of two ATP molecules and also two NADH molecules per glucose molecules going through the glycolysis. The NADH molecule can now enter the electron transport chain to produce more energy in the form of ATP through process of oxidative phosphorylation. The process of glycolysis is regulated by two major irreversible reactions. The first reaction, which is basically the phosphorylation of glucose by hexokinase, and the conversion of fructose 6 phosphate into fructose 1,6 phosphate by phosphofructose kinase 1. These play an important role in the regulation of glycolysis. Thank you.